All right, this independent variable in the enzyme investigation will measure the effect of changing the concentration of our enzyme, which is from the red potatoes, and it's called catalase. So go ahead and add the contents of the small test tube, which will be the catalase, to the large test tube, and as soon as that's done, the timer will start. So for this test tube A, we're using 20% concentration of the potato catalase enzyme diluted with distilled water. For test tube B, we're using 40%. So 2 milliliters of catalase instead. And then for test tube C, we've got 60% of the catalase enzyme. Test tube D, we have 80% of the catalase enzyme. And test tube E has the full amount of catalase with no distilled water dilution. So that's the full 100%. So you can see the reaction happening between the catalase and the hydrogen peroxide that creates the oxygen gas bubbles. And after 4 minutes is complete, we'll use a ruler and you can go ahead and see how much foam has been produced in each of these test tubes. All right, the 4 minutes is now up. So in quick succession, go ahead and look at the ruler and measure from the bottom of the oxygen gas white foam to the top. Remember not to measure from the bottom. We're not measuring from the bottom of the test tube, but from the bottom of just the foam. We're just measuring how much oxygen gas was created as product. So go ahead and measure test tube A. Remember, test tube A has our lowest concentration of enzyme, 20%. And then measure test tube B, which has 40% enzyme concentration. And then test tube C, which has 60% catalase enzyme concentration. Test tube D has our 80%, almost full concentration. And you can see then with test tube E has the full 100%. It's actually going over the top of the test tube, but you can still see where the top of that foam is in order to write down your measurement.